Hi, everybody. This is going to be our review session for chapter 8. So this is the genetic engineering chapter. Again, these don't usually last too long, so just bear with me and we'll go over the basics of what this chapter taught us. To begin with, talking about risk. So first of all, we had to kind of understand the concepts of DNA. And so if you are not that comfortable with the genetics chapter that we went over, that was chapter 6, I believe, you should probably go back and take a little bit more time on that, because there's no way that this genetic engineering chapter could make sense to you if DNA doesn't make sense to you. So that's really heavily reliant on that chapter as far as how this is going to work. And yes, genetic engineering is very important in microbiology, and most of anything that goes on in science depends upon this. So I would definitely brush up on your DNA stuff if you aren't clear on that. So having said that, restriction endonucleases, these are bacterial-derived protective enzymes which cut DNA at palindrome sequences. They leave sticky ends, and we can insert DNA that have the same sticky ends, or corresponding sticky ends that we've engineered, and stick it into those plasmids. So we cut the plasmids open. So essentially you have no, that's not the pen. Okay, so we have our plasmid, and we cut it with our restriction endonuclease. These guys operate at palindromic sequences, so the same forward as backwards. So we cut here, and then our double-stranded molecule is like this. And so if this was double-stranded, we have now created a sticky end here and a single strand sticky end there. About 4 or 5 bases that are going to stick out over those ends like that. Okay, so this is what happens when we cut with the restriction endonuclease. That's nice. So now we can engineer a gene that codes for, I don't know, insulin. So we put our insulin gene, and it has also been cut from something we engineered, cut by the same exact enzyme, which is important to remember. And it's got sticky ends. And these sticky ends, 4 or 5 base pairs, tend to match these sticky ends because we cut them with the same enzyme. And now we can put this into here. And now we have a new circle again, and it's got this little bit of new DNA in there. And that can be a gene for insulin, for example. Typically, what we use for this is going to be a plasmid as our vector. And the plasmids here, they're going to be circular. They're going to be DNA, made of DNA. And they also tend to have some sort of antibiotic resistance, so that when you put this whole mess, this whole plasmid that you've just made, you stick this guy, you stick this, and then you shove it into a new competent bacterial cell via transformation. And now you want to know which of the bacterial cells actually took up your plasmid, because they'll just grow no matter what if you put on normal media. So what we're going to do is we're going to take our plasmid that we have designed, and it's going to have in it antibiotic resistance. So if your bacteria took up your plasmid in your transformation experiment, and you played it all on that antibiotic, those ones that took up that plasma are the only ones that are going to survive on that antibiotic plate. So now you know which ones have your plasmid. And so, yeah, so that's the idea there with that. That's how restriction endonucleases can be worked with for genetic engineering. And if we're like trying to identify like a killer or something like that, if I cut your DNA with one enzyme and cut my DNA with that same enzyme, they're going to look different from one another. And so we can run those out on agarose gels, and that's going to allow us to separate them out and see by size if they are, related or not. So we'll come back to that. So CRISPR-Cas9, this is a system that's normal for bacteria as a protective mechanism for them against foreign DNA. But they can use that to cut DNA at specific sequences and potentially even alter at the germline level, so the reproductive cell level. Then we have nucleic acid hybridization. That's just using little pieces of like single-stranded DNA and seeing if it matches up with anything in on a plate or something like that. And if it does match up and it does match, then we will have a color change a lot of the time, and it'll tell us whether or not this gene is present in this sample. So that's the idea with that. Then we have polymerase chain reaction. This is multiplying DNA. We can have very, very small amounts of DNA and multiply it to millions of copies in just a couple of hours. It's a very useful tool as far as amplifying DNA samples. You can only amplify a known target. It's not magic. I can't say it enough. All right, so the steps involved in PCR, you do have to be aware of these. Heating, that's how we denature or split the strands, the DNA strands apart. Then we'll anneal primers by cooling it a little bit. Those are literally, you have to build your DNA polymerase has to add the nucleotides on to something. So that's what the primers are there for. And so we'll stick them on to the end of those primers and we'll run that polymerase out until, you've matched up everything. So we have that elongation aspect going on there. So we have denaturation, we have annealing, and then we have elongation. The elongation, this whole step actually resembles pretty closely DNA replication. All right, so that's the idea. Your polymerase that we use for this has to be special because we're going to heat things up so hot to separate the DNA strands that protein could get destroyed too. So we have a special DNA polymerase that will allow us to work even after being exposed to that high temperature. And that's from most of them that we use are Thermus aquaticus. So that is going to be a bacteria from the deep sea vents in the ocean. That's cool stuff, I think. Gel electrophoresis. So we talk about running stuff out on a gel in order to compare them based on size. This is just a gel. We're going to put it in a liquid and we're going to run electricity through it. And then that's going to push the DNA through the gel based on the charge of the DNA. And they migrate through the gel based on size, and the smaller ones go faster. Imagine that it's like, what is it, Plinko? Was it Plinko? It doesn't matter. Anyways, imagine that you are, dropping some balls down and you have all this stuff like mesh level. And you start at the top. The smaller ones are going to be able to fit through the mesh faster than the large ones. Even if the larger ones can fit, they're going to get caught up more, right? So that's what's going on with gel electrophoresis. The large ones are getting caught up more. So the large ones are going to be closer to the start, the well where you put your sample in. And then the small ones are going to be closer to the actual finish, the bottom of the gel. Okay, so smaller, bottom, larger, top. Smaller fragments run faster. All right, so cloning. So DNA can be introduced into host organisms via plasmids, which is what we were just talking about, or we can use viruses as a means to carry our genes around. Remember, bacteriophages are viruses for bacteria, so those can inject genes into the bacteria for us. And we can even have artificial chromosomes that we can design to have specific genes. Usefulness for genome sequencing helps us understand mutations and the effects of those mutations on the human body. And then we have single nucleotide polymorphisms. That would be like a change in one single nucleotide that had some sort of effect. Sometimes no effect, but like the ones that do have an effect are the ones we're more interested in, right? So one nucleotide can cause a change in one amino acid. So that's no surprise there. But that change in that one amino acid can change the entire shape of the protein and make it function differently or not at all. So that is possible. GMOs are the same thing as transgenic organisms. These are organisms that have genes from other organisms to enhance them somehow, either for production of some sort of product or for studying of that product. So then we have the concept of gene therapy, that is using viral latency to insert functional genes into humans, as well as as well as incorporating those into our somatic or our germline, depending on when in your life we are treating you or how we are treating you and things like that. There are some gene therapies that are available for some diseases. 13 now have been approved for the use of gene therapy for the treatment of those diseases. And those become permanent treatments for these diseases as well. Since they get incorporated into the cells and stay there, you don't need to receive any more treatments, ideally, once we get the genes to stick. So that's the idea of that. So somatic, ones that affect your regular, just your body cells that you're living in right now, those are going to be called somatic cells. Your germline, if it affects germline, then that is talking about your sperm or your egg or embryo, but it's going to be heritable. So you can actually pass that on to your offspring. So you are no longer passing on what mutation you did have in your genes. You just flat out don't have it in your genes anymore. You can't even pass it on to your kids. So I think that's pretty cool. So that is chapter 8, and that is the chapter 8 review. And I'll see you guys for the next one. Bye.